Hello everybody, I'm going to present results on the role of valency on the ability of ICAM1 targeted nanoparticles to effectively cross the blood-brain barrier and deliver therapeutic enzymes in cellular and animal models. First, a few words on the background. Valency is the number of targeting moieties presented on the surface of nanocarriers. Valency is known to influence the transport of nanocarriers across the blood-brain barrier, so the brain endothelial cells, and that transport might be either endocytosis, so the uptake of the carrier into terminal vesicles, such as lysosomes, or crossing the barrier into the brain, a transport known as transcytosis. Although we know of the influence of valency on that transport, the mechanisms are still unknown. We were preparing 3 different nanoparticle preps with 3 different valencies to study that mechanism. And the number of anti-ICAM molecules on the surface you can see here. And we termed them low, intermediate, and high valency nanocarriers, anti-ICAM nanocarriers. We found a bell-shaped relationship between the valency and the transcytosis efficiency, which might seem counterintuitive, because you might expect higher valency to render higher transcytosis efficiency. On the other hand, we found an inverse bell-shaped relationship of valency and lysosomal trafficking. This is because transcytosis is a complex multi-step process, where in the apical side, you have binding and uptake of the particles, which show a positive correlation with valency, meaning that the high valency particle has very high binding and uptake due to its many numbers of targeting moieties, and the low valency particle has a limiting step here because it doesn't bind as readily as the high valency particle. On the basolateral side, on the other hand, the transcytosed particles need to detach from the cell to successfully complete the process. Here, it makes sense that we found an inverse relationship, a negative correlation of valency and exocytosis efficiency, because as you can see here in the cartoon, high valency particles have a lot of connections to the cell and therefore might not readily detach or as readily detach from the cell as compared to the low valency particles. Looking at the whole cartoon, you can see that the intermediate particles might be the best compromise to render the highest efficiency for transcytosis. We then delivered a therapeutic enzyme, acid-sphingomyelinase, which is important in lysosomal diseases such as Niemann-Pick disease, by coating it onto the carriers and delivering it to the brain. So we were injecting it intravenously into wild-type mice. And in the top panel here, you can see that 30 minutes post-injection, that the particles are mostly located in the brain vessels. Whereas 3 hours post-injection, they penetrate the brain already by transcytosis and are more or less evenly distributed throughout the brain. This transport, as you can see in the lower left panel, is specific for ASM that is coated onto targeted nanocarriers as compared to free ASM or ASM coated onto carriers that are not targeted, so they were coated in IgG. We then compared the delivery of the intermediate and the high valency carriers to 3 different organs, and you can see here that liver and spleen, which are associated with passive accumulation of particles, show equal delivery or diminished delivery of the intermediate valency particles as compared to the high valency particles, which would make sense because the particles are exactly the same except for their targeting valency, meaning that passive diffusion should not be affected. When then looking at an organ as the brain, which is the organ of interest for us, where the accumulation is happening by active transport, and you keep in mind what I was just telling you, you can see that the intermediate valency particles show 100, more or less, a little more than 150% of the efficiency of the delivery efficiency as compared to the high valency particles. When then quantifying how many molecules of ASM, of the therapeutic enzyme, we were delivering to the brain are actually delivered to the brain, you can see here that intermediate valency particles deliver significantly more ASM to the brain as compared to high valency particles. If you want to know more about that, I'll be happy to answer your questions when you visit my poster. Thank you.